Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss allosteric regulation. Just as a reminder, in the previous videos, we discussed how we can change the catalytic activity of existing enzymes, how we can regulate enzymes by sequestering enzymes in compartments, by changing the availability of cofactors that are required by some enzymes, and today we shall focus on allosteric regulation. Remember that proteins work by binding ligands, some sort of chemicals, at their active site. So we have a protein, and this is the active site of the protein. If the protein is an enzyme, then the ligand is a substrate, and the function of the protein would be catalysis. If the protein was a transport protein, then the ligand would be a chemical to be transported, and the function of the transport protein would be transport. If the protein was a receptor, then the ligand would be a signal, and the function of the receptor is to carry out cell signaling. And if the protein was an antibody, for example, then the ligand would be called an antigen, and the function of the antibody would be to elicit an immune response. Now, in allosteric regulation, so in allosteric proteins, not only do we have the active site where the ligand binds, but we also have another site that is called the allosteric site. This could be one or more sites. So remember, these proteins could have one or more active sites and one or more allosteric sites. So in allosteric regulation, the binding of another chemical that we call modulator at the allosteric sites will cause a change in the shape of the protein. So when this modulator binds to the allosteric site, the protein shape will change, and that change in the shape of the protein could either increase the protein function or it could decrease the protein function. So let's look at an allosteric enzyme. Let's look at the typical structure of an allosteric enzyme. Now I use the word typical because a biochemical concept cannot be generalized. We can quite often find exceptions to the concept. So not all allosteric enzymes will have this typical structure that we'll talk about. There are exceptions. So a typical allosteric enzyme has one or more catalytic domains or one or more catalytic subunits. Each of these catalytic domains would have an active site. So there will be one or more active sites present on the protein where the substrates will bind. Since this is an allosteric enzyme, so not only will it have the active sites, it will also have domains or subunits where the modulator binds. Okay, so this is the allosteric site. So one or more regulatory or allosteric sites where the modulator binds. Since a typical allosteric enzyme has more than one active site, most often we see more than one active site, so it's not surprising, as we discussed in earlier videos, that most of these allosteric enzymes will show sigmoidal kinetic curves instead of hyperbolic kinetic curves. Let's see how the allosteric enzymes are regulated. Here's the basic concept. These allosteric enzymes can either exist in their T state or 10 state, which is the less active form of the enzyme. So there is less substrate bound, less product form because this is a less active form of the enzyme. Or the enzyme can also exist in the relaxed state, R state, which is the more active form of the enzyme. So here we have a stronger substrate binding and therefore more product is formed for the R state. Now, if other chemicals are present around the enzyme, these chemicals are called modulators. So if modulators are also present around the enzyme, then the enzyme activity could either go up or the enzyme activity could go down. If the modulator is an inhibitor, the modulator will bind to the allosteric site on the enzyme, and that binding of the inhibitor to the enzyme will stabilize the T state of the enzyme, the less active T state of the enzyme. So when an inhibitor binds the enzyme will be present in the less active T state, and therefore the enzyme activity will decrease. And this decrease happens very quickly, within minutes to hours, and therefore it's a very nice short-term regulation of enzymes that happens within cells. Now, on the other hand, if the modulator was an activator, then when that binds to the enzyme, it will stabilize the more active R state. So in the presence of an activator, most of the enzyme will be found in the more active R state, which means the enzyme activity will now increase. So here's the big picture with regards to the allosteric regulation of an enzyme. Number one, as mentioned earlier, most allosteric enzymes have one or more regulatory or allosteric sites besides the active sites. So here R represents the allosteric site, the regulatory site, and C here represents the catalytic site, which will be your active site, where the substrate binds. Now, an activator is a positive modulator. It will increase the function of the enzyme, and the function of the enzyme is catalytic activity. So positive modulator or an activator will increase the function or the catalytic activity of the enzyme. How does it do that? As we mentioned, the activator stabilizes the R state, the more active state of the enzyme, and this will cause the kinetic curve to shift to the left. So in the presence of an activator, the kinetic curve will shift to the left. That means the K0.5 instead of Km, because this is a sigmoidal curve, we can use the term K0.5. So there is a decrease in the Km or K0.5, and when there's a decrease in K0.5, there's an increased function, increased catalysis. So a shift to the left means the K0.5 will decrease and the activity of the enzyme will increase. On the other hand, inhibitors are negative modulators. Inhibitors will decrease the catalytic activity of the enzyme. And how they do that? Inhibitors stabilizes the T state, the less active T state. That will cause the curve to shift to the right. So now the curve will shift to the right in the presence of the inhibitor. And this will now increase the Km or K0.5, and that will lead to a decreased catalytic function of the enzyme. Now let's use an example to understand how a shift in the enzyme kinetic curves, either to right or left, will affect the reaction velocity. So let's say this is this enzyme here at a substrate concentration present in the cell. This enzyme is working at 60% of its maximum velocity, 60% Vmax. Now let's say the enzyme is surrounded by some inhibitor. Now the inhibitor will bind to the enzyme, and now the curve is going to shift to the right. The curve is going to shift to the right. So now we see the curve shifting to the right. So we have this new curve in the presence of inhibitor. Now at the same substrate concentration, you can see now the enzyme is working only at 10% Vmax. So from 60% Vmax in the presence of inhibitor, the enzyme is now only working at 10% Vmax. That's how the velocity of the enzyme is decreased. In the presence of the activator, the curve will shift to the left. So here now we will shift the curve to the left. We look at this blue curve, go back to the same substrate concentration as before, and if we extrapolate it in this diagram, we see now the enzyme is working at 100% Vmax. So from 60% Vmax in the presence of activator is now working at 100% Vmax. In the presence of inhibitor, it works only at 10% Vmax. So clearly the presence of inhibitors or activators around the enzyme can make the enzyme function, the catalytic function of the Vmax, go up or go down. So this is the basic concept of allosteric regulation. Now let's look at a few examples, and we'll pick the metabolic pathway called glycolysis to explain allosteric regulation. Recall from the previous videos that glycolysis, basically we are taking glucose and we are converting it into pyruvate. So we are making pyruvate, and this is the first part of what we call main street. Then the other parts were the pyruvate dehydrogenase complex, the Krebs cycle, the electron transport chain coupled to oxidative phosphorylation, and all these parts we call this the main street. Why? Because this is one of the most important metabolic pathways in the cell, because glucose is being used as a fuel to generate energy, ATP. Energy generation, as we mentioned many times in previous videos, energy generation for cells in our body is very important. The most important metabolism or metabolic pathways that happens in our cells. So let's first look at the enzyme phosphofructokinase 1 in glycolysis. This enzyme catalyzes reaction 3. This is an irreversible reaction, only goes in one direction. And in this reaction, you can see fructose 6-phosphate reacts with ATP to give you fructose 1,6-bisphosphate and ADP. Okay, so fructose 6-phosphate and ATP are the reactants, and fructose 1,6-bisphosphate and ADP are the products. This is the most important step for regulation of glycolysis. This is the committed step for glycolysis. So glycolysis regulation is done predominantly here, the most important step. Let's look at the structure of one form of this PFK, and this is the liver form of phosphofructokinase. This enzyme is a tetrameric enzyme. That means it has 4 subunits, 1, 2, 3, 4 subunits. Each subunit has one active site, so therefore this enzyme has 4 active sites, and each subunit has one regulatory domain, so therefore this enzyme has 4 regulatory sites. So this PFK1 is a tetrameric enzyme. As mentioned before, the allosteric enzymes can exist in 2 states. So PFK can exist in the R state, which is the more active form, or the T state, which is the less active form. Now the activators for this enzyme that stabilize the R state include ADP, AMP, fructose 2,6-bisphosphate, as well as several other activators. And the inhibitors of this enzyme, which will stabilize the less active T state, include citrate, phosphoenol pyruvate, as well as ATP. Did we just say ATP? Yes, well, ATP is a substrate for the enzyme, right? ATP is a substrate for this enzyme. But here we are also saying that ATP is an inhibitor of the enzyme. So how can ATP be a substrate as well as an inhibitor of the enzyme? So let's look at this problem. Now, when ATP concentration is low, that will only bind the active sites. So at low ATP concentration, the ATP will bind the active sites. When the concentration of ATP is low, it will bind to the active site and act as a substrate. However, when the concentration of ATP is high, it will not only bind the active site, but now it will also bind the allosteric sites. And when it binds the allosteric site, this high ATP concentration will stabilize the T state, the less active form of the enzyme. So more and more of this enzyme becomes less active, and the enzyme activity will go down. Since glycolysis is an important metabolic pathway for producing energy, this enzyme is closely related or associated with the energy state of a cell. So when there is low energy state of a cell, that means a lot of ATP has been used up. When we use up ATP, we usually break it down into ADP and or AMP, so the concentration of ADP, AMP will be high. These are activators of phosphofructokinase 1. Activators will increase the activity of phosphofructokinase 1 and therefore increase glycolysis. However, if the cell had high cellular energy, that means has a lot of ATP, the cell has a lot of ATP, has not used up the ATP, so the concentration of these ones are not very high, ADP and AMP, but the concentration of ATP is very high. Now ATP is an inhibitor, so ATP will bind to the phosphofructokinase 1 and inhibit the enzyme and thereby decrease glycolysis if the cell has too much energy. Note that one of the activators for this enzyme is this chemical here, which is fructose 2,6-bisphosphate. Now remember the product of this enzyme is fructose 1,6-bisphosphate. But here we are talking about fructose 2,6-bisphosphate being an activator of this enzyme. So when there is a high amount of fructose 6-phosphate, large concentration of fructose 6-phosphate, then what happens is there is another enzyme called phosphofructokinase 2, PFK2. Now this enzyme converts fructose 6-phosphate into fructose 2,6-bisphosphate. Remember, PFK1 converts fructose 6-phosphate into fructose 1,6-bisphosphate, but PFK2 will convert fructose 6-phosphate into fructose 2,6-bisphosphate. So the concentration of fructose 2,6-bisphosphate increases, and this chemical is an allosteric activator of phosphofructokinase 1. So fructose 2,6-bisphosphate allosterically activates PFK1, and that increases the rate of glycolysis. So in this case, the substrate of PFK1, which is fructose 6-phosphate, this chemical here, is actually indirectly stimulating PFK1 through the action of PFK2. This activation of an enzyme by its own substrate is called feed-forward stimulation. Now let's look at another glycolysis enzyme, and this is pyruvate kinase. Pyruvate kinase catalyzes the irreversible step of glycolysis, which is reaction 10, shown here. The reaction is between phosphoenol pyruvate and ADP. The products that are formed include pyruvate as well as ATP. There are several different isoforms or types of pyruvate kinases present in our body. Pyruvate kinase M1, for example, is found predominantly in muscle, brain, and heart. PKM2 is found in various tissues, including kidneys, adipocytes, pancreas, lungs, as well as in rapidly dividing cells such as fetal tissue, and interestingly, in cancer cells. Pyruvate kinase L, PKL, is rich in the liver, pancreas, kidneys, and enterocytes, while red blood cells predominantly contain the isoform called pyruvate kinase R. So what's the structure of pyruvate kinase? Let's focus, since there are several different types of enzymes, of isoforms, so let's focus on this one here, pyruvate kinase M2. Now this enzyme is found in the tetrameric form, 4 subunits. It can also be found as a dimer, 2 subunits. The dimer is nearly inactive, has slight amount of activity, or it can also be found as a monomer, and the monomer is inactive, doesn't have any activity. As mentioned before, this is an allosteric enzyme, so it can exist in its more active R state, as well as the less active T state. Activators will stabilize the R state, and these activators include fructose 1,6-bisphosphate. So this intermediate here is actually an activator of pyruvate kinase. This is another example of feed-forward stimulation. Fructose 2,6-bisphosphate is also an activator. AMP is an activator, stabilizes the R state, as well as the amino acid serine has also been found to be an activator of this enzyme. Inhibitors of this enzyme are several. That includes ATP, since ATP is a product of this reaction, as well as a product of glycolysis in general. We can call this product inhibition. Alanine is an inhibitor. Actually, alanine can be made from pyruvate. As we saw in starvation, pyruvate converted to alanine by transamination. So alanine is also an inhibitor. Another amino acid, phenylalanine, is also an inhibitor because it stabilizes the tetrameric T state. The thyroid hormone T3 is also an inhibitor because it stabilizes the monomeric form of this enzyme, which is an inactive form of the enzyme. And finally, let's look at hexokinase. So we looked at phosphofructokinase, which catalyzes this irreversible step, number 3. Pyruvate kinase, which catalyzes this irreversible step, number 10. And now let's look at hexokinase, which catalyzes this irreversible step of glycolysis, number 1. So in this reaction, what happens? Glucose reacts with ATP to give us glucose 6-phosphate and ADP. This is the reaction that is catalyzed by hexokinase. Once again, this enzyme can be found in as many different isoforms. There's hexokinase 1, hexokinase 2, hexokinase 3, and then we have this very important hexokinase 4, also called glucokinase, which is found in the liver cells as well as the beta cells of the pancreas. And these two organs are very important in maintaining our blood glucose, because as you know, beta cells of pancreas secrete insulin, and of course liver plays a very important role, as we saw in starvation and the what happens after a meal in previous videos. The liver plays a very important role in maintaining blood glucose. Now, here's something very interesting. Hexokinase 1, 2, and 3 show hyperbolic curve. Okay, hyperbolic curve, because it has only one active site. So in general, we mentioned if you have only one active site, we see a hyperbolic curve. However, hexokinase 4, which also has one active site, actually shows sigmoidal kinetics. So hexokinase 4, so although all these isoforms have one active site, hexokinase 4 shows sigmoidal and not hyperbolic. So what is the reason for this phenomenon? And we shall see the answer is that hexokinase 1, 2, 3 exists in only two states, what we call the closed state and the open state, while hexokinase 4 can exist in three states: the super open state, the open state, and the closed state. So let's look at how this phenomenon is explained by the fact that hexokinase 4 can exist in three different states instead of two different states. So here's hexokinase 4, glucokinase. Notice there's only one active site. So there's one protein, a big protein with only one active site. But there are three forms of this enzyme that are present. There is this closed form, which is active. Then there is the open form. And then there is the super open form that is inactive. So let's see how this enzyme being present in these three possible forms explains the properties of this enzyme, of glucokinase. What are the properties? Glucokinase has a very low affinity for glucose. That means the Km for glucose for this enzyme is very, very high. The other property is that this enzyme shows a sigmoidal curve. And a third very important property of this enzyme that is present in the liver and the pancreas is its ability to regulate our blood glucose levels. So this is one of the models that have been put forward to explain these properties of glucokinase. So here we have the super open form, the inactive form. This is thermodynamically the most stable form. So when there's no glucose present, the enzyme exists in this state. But once the substrate, glucose, is present, now very slowly, this is a slow step, the super open form is converted into the open form. You can see the glucose bound to the active site. Once the open form is formed, ATP binds, and very quickly, this is a fast step, the enzyme changes from the open form to the closed form. Now the closed form is the active form. So with glucose and ATP bound, the reaction occurs. So the end of the reaction would be the formation of products, glucose 6-phosphate and ADP, and the enzyme converts from the closed form where the reaction occurs, it goes back into the open form, into this form. And in the open form, the products glucose 6-phosphate and ADP are released. Now, if the glucose concentration is high, there's a lot of glucose around, then what happens is this open form will basically enter this, get converted to a closed form. This is called the fast cycle. Okay, so in the presence of high amounts of glucose, we have the fast cycle being used by the enzyme. However, when there is very little glucose or no glucose available, then this open form of this enzyme will go back to the most stable form, which is the inactive super open form. This is called the slow cycle, because for this form to be active again, it goes through the slow step. So this enzyme therefore can follow the fast cycle or the slow cycle. In the presence of high concentrations of glucose, the fast cycle is followed. When glucose concentration is low, the slow cycle is followed. And this explains the sigmoidal curve. The sigmoidal curve. At low concentration, the activity is low, and then as the concentration rises and rises, the enzyme activity increases tremendously. The sigmoidal curve. This also explains why the liver and the pancreas will take up more glucose only if the blood glucose concentration is high. Because when the blood glucose concentration is high, this enzyme is more active. It will convert the glucose to glucose 6-phosphate. But when the blood concentration is low, this enzyme is not active and therefore will not convert glucose to glucose 6-phosphate. What about allosteric regulation of this enzyme? In the presence of inhibitors, so one example of an inhibitor of this enzyme is glucokinase regulatory protein, or GKRP. The inhibitor stabilizes this super open form or the inactive form, and therefore the enzyme activity goes down. Now, in the presence of activators, there are several drugs that can act as activators for this enzyme because this enzyme plays a very important role in controlling blood glucose concentration, therefore very important in diabetes. So in the presence of activators, the open form is stabilized. This form is stabilized. So now the enzyme has only the open and closed forms available, very similar to hexokinase 1, 2, 3. So now the enzyme uses only the fast cycle. And therefore, in the presence of activators, the enzyme no more shows the sigmoidal curve, it actually shows a hyperbolic curve because it only has, in the presence of activators, only these two forms. Just like hexokinase 1, 2, and 3, the open form or the closed form. It does not have this super open form. Allosteric regulation is very common in metabolism, and you can see that in many metabolic pathways besides glycolysis. There is allosteric regulation in the PDH reaction, Krebs cycle, gluconeogenesis, glycogen breakdown, and so on, fatty acid metabolism. And we shall look at these examples when we talk about regulation of metabolism after the postprandial state, as well as during starvation in future videos. In the next video, we shall discuss covalent modification, another very common way to regulate enzymes. So until the next video, once again, thank you very much for watching this video and supporting our channel. Goodbye, have a wonderful, wonderful day, and hopefully we'll see you soon.